Hello everyone, my name is Nuran Shalma. I'm a senior medical student at Tanta University and I'm a MMR schooler. I'll be presenting blinding in clinical trials. What is blinding? How to blind? What is the importance of blinding? How unblinding may occur? Why not blind and what to do when blinding is difficult or impossible? So let's start by knowing what is blinding. Blinding, sometimes referred to as masking, is a process which ensures that the participants, the investigators, and the staff, they don't know if the participant is in the intervention or in the control group. So, how do you think we can blind them? Patients in the control group receive an identical placebo pill, patch, or injection. And if the intervention is a surgical procedure, then they will undergo an identical sham procedure. So, what is the importance of blinding? If we already did randomization, then we already minimized the differences between the two groups at baseline. We want to keep these differences minimized through the follow-up, and here blinding becomes important, as it minimizes the co-interventions, the biased outcome ascertainment, and the biased measurement of the outcome. And now let's talk about co-interventions. Co-interventions are medications, therapies, or behavior other than the study intervention, and these could change the risk of developing the outcome of the interest. So let's take an example. Suppose we're running a trial testing aspirin versus placebo, and our outcome is the cardiovascular events. What if the patients in the intervention group, and they really believed in our intervention, decided to take extra drugs, they decided to be healthier, they decided to do more exercise? This would be an example of co-interventions. But what if the intervention didn't come from the patients themselves? What if the study staff, who really believed in our intervention, decided to advise the patients to do more exercise, to eat healthier, or to take extra medications? If this happens non-differential, so they, for an example, advise both the intervention and the control group, let's say, to do more exercise, and that would change the risk of developing our outcome. If this happens non-differential, this would decrease the power. However, if it happens differential, meaning that our staff just advised one group, not the intervention and the control, only one group of them, this would cause bias. And now let's talk about the biased outcome ascertainment. If the group assignment is known, then the participants may report symptoms or outcomes differently, and the physicians or the investigators may elicit symptoms differently. And this becomes problematic with soft outcomes, such as chest pain, disability, and satisfaction. So let's take an example. Suppose we're running an unblinded trial testing statin therapy. In here, the investigators may be more likely to ask the participants taking the intervention about muscle pain or tenderness. They are more likely to order tests from them for them to make the diagnosis of myositis. And now let's talk about the biased outcome adjudication. Outcome adjudication is when the study staff is the one who decides if a change in the outcome has occurred. So, he may classify events differently, knowing which patients were in the intervention group and which patients were in the control group. And this is problematic with soft outcomes. So, let's take an example. In the Canadian Cooperative Multiple Sclerosis trial, 165 patients with multiple sclerosis underwent plasma exchange, cyclophosphamide, and prednisolone in the intervention group, while the control group underwent sham plasma exchange, and placebo meds. The severity of multiple sclerosis was assessed using a structured examination, once by blinded and another time by unblinded neurologists. And the fact that they were blinded or unblinded just affected their judgment. More improvement with plasma exchange was found by the unblinded neurologist, but not the blinded ones. And now let's move on to some terminology. A single blind means that the participants are not aware of the treatment group. A double blind means that both the participants and the investigators were unaware, and a triple blind may have an unclear meaning, as the third person who was blinded may be the one who performed the test, it may be the outcome adjudicators, or it may be the safety monitoring group. So, how unblinding may occur? Blinding is difficult even for drugs. An identical placebo is difficult to prepare, as the drug may smell, taste, or feel different. And even if we manage to prepare an identical placebo, the drug may cause side effects. The test results may unblind, or the participants themselves may test the drug to know in which group they are. So, knowing that blinding is really important. Why might we not blind? It's difficult to blind the participants if they are assigned to surgery, exercise, diet, or education. So, what are we gonna do? If blinding is difficult or impossible, we have to be clever, courageous, measure the degree of unblinding, and just do the best you can. Be clever. Researchers used garlic for cholesterol lowering, so they prepared an odorless, tasteless garlic preparation, and they prepared an identical placebo. Be courageous. The internal memory ligation procedure was used to relieve angina. However, after blinded trials were conducted, where patients in the control group underwent a superficial skin incision without actual ligation of the internal memory artery, no significant difference was found. Similarly, in the orthoscopic debridement of the knee joint, which is a procedure used for relieving pain from osteoarthritis, patients reported an improvement of about 50 to 60% in their pain scores. However, after blinded trials were conducted, where in the control group the arthroscope was inserted into their knee joint, but without actual debridement of the knee cartilage, no significant difference was reported in their pain scores. Measures the degree of unblinding. In trials that are partially blinded, you can ask the participants and the study staff to guess the treatment, and if unblinding is substantial, you can assess its impact in the discussion. Do the best you can. Find investigators and staff who measure the outcomes, minimize differential co-interventions, preferred hard outcomes as death, and prefer measurement over self-report. So, as an example, prefer myocardial volume oxygen over self-reported exercise ability. And now let's summarize. Blinding is as important as randomization. It maintains the comparability of the groups during the trial, and it minimizes the bias from co-interventions, biased outcome ascertainment, and adjudication. And we're done. Don't forget to follow Mars on LinkedIn, Twitter, Facebook, and the site, Mars Arabia. Thank you.